My lab studies the circuitry in the spinal cord that controls movement. In this video abstract for a new study in neuron, Stephanie Koch, the lead author, will describe work that she's done to identify a class of inhibitory neurons that control sensory transmission during movement. This work was first started by a former postdoc on my lab, Marta Garcia del Barrio. When Marta first joined the lab, she became interested in cells in the dorsal spinal cord that are important for transmitting and gating sensory information that's coming in from the periphery. This sensory information that comes from muscles and it comes from the skin is known to be important for the dynamic control of movement. Marta actually became particularly interested in one population of neurons. These are a class of inhibitory interneurons that are marked by the expression of a transcription factor, RORβ. One of the reasons that Marta was particularly interested in these cells was it was known from former studies in the cat that sensory transmission is strongly regulated during movement, and in particular it is attenuated during the swing phase of stepping. What was not known was the identity of the neurons that might be responsible for this and what happens when this functionality is lost.
So my name is Stephanie Koch and I'm a postdoc in Martin Goulding's lab. And when I joined the lab, I was introduced to this project, and this project is particularly interesting because of the phenotype that you see in RORβ null mice. And what's interesting about them is that if you knock out this gene globally within a mouse, they have this fascinating locomotor phenotype, which is called a duck gait. And what you see is that these mice display a hyperflexion during the swing phase selectively, and that's very task specific. So Marta had been doing all of her analysis using an antibody, and I decided to try to look into these cells with a little bit more detail using a Cree line made by the Allen Brain Institute, where they inserted Cree into the RORβ locus. And when I did that and started to analyze these cells in a little bit more detail, I found not only these lamina 3 located RORβ interneurons, but also ones a little bit deeper in lamina 5. And those are particularly interesting for us in the lab because they're well situated to receive information from the muscles, proprioceptive information, but also potentially to gate that information. So that really gave us this idea that maybe it was these lamina 5 RORβ expressing interneurons that were key for controlling this fine movement that we see normally. So at this stage, we really wanted to verify that it was the spinally located RORβ interneurons that were necessary for this duck gait that we saw. And we did this by a number of genetic methods, both by ablating the cells that we saw in the spinal cord, as well as silencing them or just preventing the function of the gene within an inhibitory interneuron. And that confirmed that the cells that we saw, the inhibitory cells that we saw, were really necessary for generating this duck gait phenotype that we saw in the RORβ null. So what we tried to do next was try to find out a little bit more about how these spinal circuits in the RORβ inhibitory mutants were activated whilst they were stepping. And the way that we decided to do this was to use electrophysiology and just look at the isolated spinal cord. And what we found was that the motor circuits of these RORβ inhibitory mutants were activated much more easily to any kind of sensory input than their controls. And that really pointed towards this idea that there was maybe this lack of presynaptic inhibitory control. To be able to probe that directly, we used optogenetics also in the isolated spinal cord preparation with electrophysiology. And we found that these interneurons themselves make direct contacts onto both proprioceptors and to low threshold myelinated afferents. And that really closed this circuit for us and showed us that these interneurons, not only are they in the right place, and not only are they able to make the right contacts, they also functionally do inhibit these afferents and then make this very tight circuit that we would expect them to make during walking movements. I think what's really exciting about this project is that we've managed to really isolate and describe this very local and very specific circuit. And the circuit is only activated during stepping, so it's very task selective. What it does is it's able to presynaptically inhibit information coming from the muscles and from the skin so that we can really focus all of our attention to normal stepping.